Okay guys, let's start. So first of all of you are welcome in this 2-day lecture series on advanced biology techniques, and it is organized by Department of Microbiology and Biotechnology, Patkar College of Arts and Science, and Varde College of Commerce and Economics with the Association of Logical Biology. And I'm very thankful to the organizing committee from the college, specifically to the principal Dr. Shrikant and Ruchita ma'am, because she was the bridge between logical biology and the college, and also to Anupama ma'am. So in these 2 days lecture thing, we are going to discuss about flow cytometry, secondly about the world of animal cell culture, or specifically the human cell culture, and quantitative real-time PCR based gene expression analysis. Generally what happens that people discuss about the data and data sets and analysis, but I have kept my presentation with good fundamentals which will be easy to understand, and after these lectures you will be able to understand the basics of these techniques, also what kind of experimentation you can do, what are the importance of these techniques, as well as what kind of applications are of these techniques which you can apply in your research, current research, or in future you want to be a researcher. And I will also be giving you importance about learning the techniques, why it is important nowadays. And for the students, I am having 5 to 6 advices, and these advices I will be giving in between the lectures. So after every 15-20 minutes I will come up with the advice, which if you are interested you can follow, and you can really make a good career in the field of research biology. So in these 2 days schedule, today I am going to mainly discuss about flow cytometry, multicolor flow cytometry and its applications, and tomorrow we are going to discuss the cell culture and real-time PCR based system. So let's start with the discussion. So if somebody gives a word cytometry, then the statement which comes in my mind that cytometry is nothing but measuring the physical and chemical properties of a single cell. So you have taken a single cell and you really want to measure different type of physical and chemical properties, presence of biomolecules, shape, size, density. All these things you are measuring under the branch of cytometry. And when I add the word flow in front of cytometry, it becomes flow cytometry. And you are doing the same thing, measuring the physical and chemical properties of single cell. But now you are utilizing a fluid stream. You are utilizing a flow of liquid in an instrument which is called as flow cytometer. So the word itself is not a complex word. It's a good system. You are going to utilize a flow system to generate single cells, and there your instrument is going to analyze those single cells, and we'll be measuring some chemical properties, we'll be measuring some physical properties, and the whole thing is called as flow cytometry, and we call the instrument as flow cytometer, right? So I always believe that whenever you are learning the technique, you are learning about a technique, then it is not about how you are going to switch on or switch off the system, but it is about, are you understanding the basic fundamentals on which the instrument is working, and mainly are you learning what kind of applications and what kind of experimentation you can do on that technology, and are you able to design the experiments on your own, and finally are you able to analyze the data. So the most important thing is analyzing the data, right? So we are not interested that much in the electronics of the instrument itself, that what are the electronic chips, how the detectors are working, what are the photomultipliers and all. We are interested in the basic principle and then experimentation types and the data analysis. Now we do one simple experiment on flow cytometry. What are the steps? What I can do, I can take a tissue or I can take cells. Okay, if I am taking a tissue, then I will degrade the tissue by using some protease enzymes, and from this tissue I will be taking single cell suspension, right? So ultimately we require a cell suspension in a buffer to do flow cytometry. That cell suspension can directly come from the cultured cells, from commercially available cell lines, even from the bacterial cultures, right? Or it can come from the tissue. So we can take whole tissue and we grind it, we treat it with some enzymes, and then we get what? Single cell suspension. This suspension you put in a cuvette or in a tube, right? So you are having tissue or directly single cells. Then you are putting those cells in the tissue, those cells in a test tube or in a tube or in a cuvette, and that tube you put in the flow cytometer. That is your work. You take the cells, prepare a single cell suspension, put them in a test tube, and the whole tube you are going to put in the flow cytometer. And after that, flow cytometer, the instrument will start its own function or its own analysis. How it is doing that? We are going to just learn it, right? So this is the working part of a flow cytometer. So the flow cytometer is taking your single cell suspension or the solution of cells, and initially those cells are present in a suspension, and this one circle is denoting one cell. So till this part you can see that cells are together with each other and they are in form of a single cell suspension. What a flow cytometer is doing, flow cytometer generates a high pressurized flow of fluid. So from this direction a fluid is coming, a buffer. From this direction a fluid is coming, again the buffer, and it is pressurizing these cells from both the direction to generate a single array of cells. Single array of cell means you are having a line of cells. So you are having first cell, then second cell, then third cell, then fourth cell, right? So cells are present in a single array after the functioning of flow cytometer. And this whole system is called as hydrodynamic focusing. So what is a hydrodynamic focusing? You take the suspension, you pressurize the suspension from both the sides, and then ultimately you get a single array of the cells where one by one cells are passing through this tube. Then finally the single cell come to a stage where it is interacting with laser, or you can say it is interacting with light. Laser is also producing some kind of light, some kind of energy in terms of h mu or hc by lambda, in terms of frequency or in terms of wavelength. Then if a cell is exposed to a laser light, then obviously it will interact with the wavelength of light, and those interactions are measured by whom? The detectors which are present in the instrument. So for me, this is the flow cytometer doing what? In first stage, creating hydrodynamic focusing and creating a single array of cells. Then second stage, your single cell is interacting with the laser, and third stage, those interactions are measured by the detector. And this is here we have learned about the instrument, which is more than enough. Now we have to learn about the chemistry and biology and the interaction of the cells with the laser light. Now instrument is having very simple function: hydrodynamic focusing, single array formation of cells, interaction with lasers, and finally the detections. Now there are new instruments coming in the flow cytometry which are not using the hydrodynamic focusing only. They are using sound waves here. They have a piezoelectric device here which can create a sound wave, and by using sound wave they can focus the cells. So there are different kind of models, but ultimately what an instrument is doing, it is giving you a single array of cell which can interact with the laser in a mode where one cell is interacting at a time, right? And the interactions are measured by the detector. Now This picture is explaining actually the interaction of cell with light in a flow cytometer. So this is our single cell which is coming from that array one by one. This is our laser light source here, and this is the incident light beam. So this light beam is going here and interacting with what? This cell, right? Now, if we are well aware of our past syllabus of physics, then we know that there will be some interaction between the cell and this light beam. And there are three types of interactions. Okay, we go through these interactions one by one, and then we will be able to understand what a flow cytometer is actually doing. So the single cell is coming here and it is interacting with a light beam. So first, this light beam or this light can have scattering. Scattering means it can collide with the edges of the cells and it can scatter. Okay, scattering means it is not following a straight line path, but it is going in some other direction. Okay, so straight line is coming at 0 degree, you can say, and then suddenly it is scattered on some angles. So if it is scattering of the light at 0.5 degree to 5 degree angle, means after the changing the path, the angle forming is between 0.5 degree to 5 degree, then this kind of scatter is called as forward scatter or FSC, right? Forward scatter or FSC. And this kind of scattering is giving us the information about the size of cell. So you can say if you are having a smaller cell, then you will be having less value of what? Forward scattering. And if you are having a bigger cell, you will be having more value of what? Forward scatter. So this is the first parameter of flow cytometry, where the light is scattering because of the size of cell and the angle measurement is between 0.5 degree to 5 degree, and it is creating a parameter which is called as FSC or forward scatter. And forward scatter is giving us the information about size of cell. Smaller size, smaller value of forward scatter. Bigger size, bigger value of the forward scatter. So as a biologist, even if you don't want to go into the details of physics, it's fine. You can just say that forward scatter is a parameter of flow cytometry and it gives the information about size of cell. More value of forward scatter means cell is big, and less value of forward scatter means cell is small. Okay. Now some of the light can enter inside the cell and there it will be scattered not by the size of cell but by the granularity of cell. Granularity of cell means the cells are having different biomolecules like proteins, lipid, carbohydrate, then cell organelles like mitochondria, lysosomes, other things, right? So it is interacting inside the cell with some granules and there it is scattering. And that scattering is generating an angle between 15 degree to 150 degrees. And this parameter is called as side scatter or SSC. So we were having forward scatter, which is giving us information about size of cell. And we are having side scatter, which is giving us information about granularity of the cell. So if a cell is having more granularity, then there will be more value of SSC or side scatter. And if a cell is having less granularity, then it will be having less value of SSC. So one parameter FSC is giving you information about size. Second parameter is SSC, which is giving you information about internal granularity of cell. Third kind of interaction can be a cell can have fluorescent factors, means it can have some proteins, or artificially you can label some proteins by utilizing antibodies. Then what will happen? These fluorescent factors can absorb this light, okay, and after absorption they can emit some fluorescence, correct? And this fluorescence can also be measured by what? Detectors. So detectors are measuring the size by measuring the forward scatter. The detectors are giving the information about granularity by measuring the side scatter, and the detectors are measuring whether a cell is containing any fluorescent factor or not by measuring the fluorescence coming out of the cell. So a flow cytometer does three basic things: measurement of size, measurement of granularity, and measurement of presence of a fluorescent molecule or a fluorescent factor inside the cell or on the surface of the cell. Size, granularity, and fluorescence. FSC, SSC, and fluorescence. These three information are given by the instrument, and we use that information to understand the properties of cells, which I am going to explain by using some beautiful examples, that which kind of data you get and what kind of conclusion you make out of those data sets or those values. So I'm again summarizing. We are measuring FSC, size of cell, we are measuring SSC, granularity of cell, and we are measuring fluorescent signals, which can be of multiple types. Like the cells can have inherent fluorescent proteins like blue-green algae, and some algal cells can produce the fluorescence. Then there are some fluorescent proteins which we can express by using genetic engineering, like GFP, green fluorescent protein, CFP, cyan fluorescent protein, YFP, yellow fluorescent protein, RFP, red fluorescent protein. So these proteins can be expressed inside the cell with the help of some kind of genetic engineering. Then we can target some proteins with monoclonal antibodies. These antibodies will be going inside the cell or on the surface of cell and will be binding to their target proteins, and these antibodies will be giving us what? Fluorescent signals. So antibody fluorescent signal means we are getting indirectly fluorescent signals from the proteins present on the cells. And sometimes we can use fluorescent dyes for staining of cell parts. We can use a fluorescent dye called as PI, propydium iodide, to stain the DNA and get the signal of DNA in the flow cytometry. Then 7-AAD, then some cell death analysis dyes are there. But ultimately we are having different types of what? Fluorescent signals. Now we are having the basic information about the cell system, by the fluorescent information about the flow cytometric system. And we know we are doing nothing with the help of flow cytometer. We are arranging the cells in a single array, and after that arrangement cells are interacting with laser. It can give you three parameters: forward scatter for size, side scatter for granularity, and fluorescent signal for the presence of protein or some fluorescent dyes. Or here comes my first advice to the students specifically, that biology is not a subject. Biology is the combination of physics, chemistry, and mathematics. And if you're really interested in making good career in the field of life science research or biological research, then it is very important to have good fundamental knowledge of physics as well as chemistry as well as mathematics, because we have seen here you are needing the knowledge of light scattering, angles. If you are not having the knowledge of that thing, then it will be very difficult to understand what biology in a good way. Then you always have to mug up. Then you always have to remember the things, because you are not having the help of fundamentals from physics, chemistry, and mathematics. And according to me, biology is not a subject, but it is the combination of physics, chemistry, and mathematics which are explaining the function, the characteristics of cells. It is very important to understand that. You will not believe that the biological polymerization reaction, like DNA replication, where we are polymerizing the nucleotides, transcription, where we are polymerizing the nucleotides, translation, where we are polymerizing the amino acids, glycogen synthesis, where we are polymerizing the glucose, starch synthesis, where we are polymerizing the glucose. These are different aspects to understand. But one thing is common in all the biological polymerization reactions. In all the biological polymerization reactions are following single reaction mechanism, that is nucleophilic substitution reaction, right? So if we are good at nucleophilic substitution reaction, then it is very easy to understand all these phenomena because we are knowing the fundamental behind that. So a good biologist is always a good physicist, good chemist, and good mathematician. So you have to take the help of other subjects. Then it is easy to understand the fundamentals of biology in the perfect way. Anyways, we go ahead and I tell you there are some fluorescent dyes which we can use to tag the antibodies, to tag the proteins, to tag the DNA and other biomolecules, and the names are FITC. These are the absorbance and emission values. Then PE, phycoerythrin, APC, L-phycocyanin, PERCP, which is the modification of PE and other combination. Then there are so many dyes which you can utilize in your flow cytometry experimentation. Then there are some fluorescent proteins, as I said, CFP, EGFP, YFP, RFP and all. So this table is just telling you, okay, you can utilize different colors of fluorescence and you can measure, you can use them in your experiment. Now, what is the best way to learn a technique? The best way to learn a technique is by doing one experiment. So obviously, online we cannot do one experiment, but I'm having a method to understand the experiment. Let's do that. Now, I have taken blood from a human, okay, and from that blood, by using Ficol gradient method or any other sugar gradient method, I have isolated PBMCs, peripheral blood mononuclear cells. So you can say these PBMCs are not having RBCs. These cells are having lymphocytes like T cells and B cells. These cells are having granulocytes like neutrophil, eosinophil, and basophils and all. They are having macrophages, they are having monocytes, they are having NK cells. Any cell which is having the nucleus comes under the category of what? PBMCs. So we have taken the blood and we have taken the PBMCs. Now, in PBMCs, we have added two types of antibodies. CD3, antibodies against a protein which is called as CD3, and antibodies against another protein which is called as CD19. We added the antibodies. We have incubated these cells for 37 degree for 30 minutes, right? And then we put the tube in the flow cytometer. What flow cytometer will do? Flow cytometer will measure the size, will measure the granularity, and will measure the fluorescent signal coming out of these antibodies because they are fluorescently labeled. So actually we are getting indirect fluorescent signal from these proteins CD3 and CD19. Now the people who don't know about the CDs, I just give a quick information about CDs. So CD is a cluster of differentiation, means these are the surface proteins present on different type of cells, okay? And by utilizing these CD proteins, we can phenotype the cells. Phenotype the cells means if somebody asks me how many types of T cells you have, then I give the answer. The basic classification is CD4 positive T cells and CD8 positive T cell. What do you mean by that? CD4 positive T cells are the T cells which are having presence of CD4 protein on their surface, right? Then CD8 positive T cell means CD8 T cells are the cells which are having the presence of CD8 protein on their surface. So these molecules, there are multiple CD molecules. I think right now there are 300 or 350 types of CD molecules which can be utilized to explain a type of cell. So somebody asked me, can you explain regulatory T cell or suppressor T cell or suppressor T lymphocyte? Then I give the answer yes, because suppressor T cells are CD3 positive, CD4 positive, CD25 high, CD127 negative, CD152 positive, and FOXP3 positive. So this is the definition of what? A T cell. Which kind of T cell? Suppressor T cell or regulatory T cell for me. Right? So what I have done here, I have taken the PBMCs and I have added fluorescently labeled antibodies against the CD3 and CD19. So what are these proteins? CD3 is a protein which is present on all T lymphocytes. All T cells are having essential presence or essential expression of which protein on their surface? CD3. And CD19 is a protein which is present on B cells. So if I want to differentiate between T cells and B cells, then I will be saying T cells express CD3 and B cell express CD19. And by looking at the fluorescent signal coming from the antibodies which are binding to CD3 and CD19, we can understand the data. So we'll put that into the test tube. I will incubate before putting them with the antibodies against CD3 and CD19. Against means that antibody is giving you, for example, blue fluorescence and binding to which protein? CD3. So if a cell is CD3 positive or a cell is expressing CD3, I will be getting blue fluorescence from it. And a cell which is expressing CD19, I will be getting what? Green fluorescence, for example, from that, right? So what kind of data, what kind of result we get from the flow cytometer? Let's see it and try to analyze it. Wow, we are getting this kind of some graph. So when I'm looking at it, it is looking a little bit complex, but don't worry about that. If we go in a step-by-step manner, then it is easy to analyze. So if I look at the first graph, right, then on the x-axis I am having FSC. So जैसे ही I listen the word FSC, the thing which comes in my mind that this axis is actually giving me the information about size of cell. Very simple, because FSC, forward scatter, is the measurement of what? Size of cell. Then on y-axis we are having SSC, that is side scatter. जैसे ही I listen the word side scatter, the thing which comes into my mind is the y-axis is giving me the information about granularity of the cell. So x-axis size and y-axis granularity. Okay. Now, based on the values of FSC and SSC, we have three types of cells. Okay, here one dot is equal to one cell. So many dots are clustering in the area. It means their size and SSC granularity is the same thing. So in this graph, I get actually four types of cells. Here, the cells which are having very low value of what? FSC and SSC, it means they are dead cell. This is the debris which is coming because of our experimental procedure. Okay. Then we are having a population here, and this is the population of what? Lymphocytes. Then we are having a small second population is there, which is the population of what? Monocytes. And then we are having this third population, which is the population of granulocytes. So why granulocytes are coming here? Because obviously they will be having higher value of granularity because they have acidic molecules, basic granules, neutral granules in neutrophils and all. So their SSC value will be automatically high. Right? So I am getting three populations: lymphocytes, monocytes, and granulocytes. Now everybody knows the use of Microsoft Paint. So what happens in Microsoft Paint, you can select something. Here also I can select like this. I can select this area and I can tell the software, okay, tell me how many cells are coming in this area. Then software will calculate and it will say you are having 50% of the cell. 50% of what? Whatever cells are present in this graph, out of them 50% are what? Lymphocyte. Yes, within 30 minutes experiment, by utilizing flow cytometry, I can measure the percentage of lymphocyte in a person's blood. I can select here and there's software will be telling me this is the percentage of monocyte. If I'm selecting here, the software will be telling me this is the percentage of what? Granulocytes. Very easy, right? So within 30 minutes, we are having exact count and the percentage of different type of cells like lymphocytes, monocytes, and what? Granulocytes, right? Very easy to understand, very easy to analyze the data. Now, I can select here and I can tell the software, okay, please show me this area into another graph. I will double click it and I will be getting this graph where on x-axis I am having CD3, it is not 5 actually, it is CD3, and on y-axis I am having CD19. So CD19 means here I am getting the fluorescent signal from the cells which are CD19 positive. Here I am having the fluorescent signal coming out of the cells which are CD3 positive. Very simple. CD19 positive here and CD3 positive here. Okay. Now I can divide the graph. I am having three populations again. You can see here one population. Again, one cell is equal to, one dot is equal to one cell. I am having here some population. Then I am having here some population. So three populations we are having. I can divide the graph into a quadrant. Right? I can just select it again as Microsoft Paint. And people who have seen the Ramachandran plot, again there are 4 quadrants. Similarly, in this graph we are having 4 quadrants. What I am doing here, this quadrant is negative for CD3 fluorescence and this quadrant is also negative for CD19 fluorescence. Okay, fluorescence, a dye has been used on the antibody which is binding to CD3, which is giving me green fluorescence, FITC, and here a dye has been used for the antibody which is binding to CD19, and that is giving me red fluorescence that comes from phycoerythrin. So we have added these two dyes on 2 antibodies, though those 2 antibodies are mixed in the PBMCs and we are getting this kind of result. Now, I am having these cells which are negative for CD3 and negative for CD19, but they are lymphocytes because I have selected this area. What do you mean by that? Then I will be saying these cells are negative for CD3, means they are not T cells. These cells are also negative for CD19. It means I will be saying these cells are not also B cells. These are double negative cells. What can be? They can be NK cells, they can be other types of cells, but they are not B cells and T cells. Now look at this quadrant. This quadrant is positive on x-axis, means it is positive for CD3 expression, but this quadrant is negative on y-axis, means this is negative for CD19. So I can say that these cells are CD3 positive but CD19 negative. So I will be saying that these cells are T cells but not B cells. So these were not T cells, B cells, and these are only T cells. So I can calculate the percentage, and here software is saying that in your sample you are having 66.39% cells which are what? T lymphocytes or T cells. Now look at this quadrant. This quadrant will be saying these are the positive for the fluorescence of CD19, but these cells are negative for the fluorescence of what? CD3. So these cells are not T cells, but they are what? B cells, because B cells are CD19 positive and CD3 negative. T cells are CD3 positive but CD19 negative. So software is telling me you are having 18.11% of what? B cells. And in this quadrant, I am having very few cells, that is 0.38. What will be this quadrant? This quadrant will be CD3 positive, because these are positive for the fluorescence on x-axis, and CD19 also positive because they are also positive for CD19. So one smart person can ask me, sir, why this quadrant is having very few cells? Then the answer is very simple. You don't have a cell which is positive for both T cells as well as B cells. You cannot have a common cell which is having the characteristics of both T cells and B cells. So it was a very basic small experiment, but by utilizing this experiment, we found that a given sample is having these many percentage of lymphocyte, then out of the lymphocyte 66% are T cells, 18% are B cells, then there are some cells which are double negative, then there are very few cells which are double positive. So within half an hour from a given blood sample, I can find out that what is the percentage of B cell, what is the percentage of T cell, what is the percentage of granulocyte, monocyte and lymphocyte. What we have done, we just have measured size, we just have measured granularity, and we have measured the fluorescent signal coming from the antibodies which are binding against CD3 and which are binding against CD19. So this was a very basic experiment you can do. Now the problem comes. What is the problem? I tell you. One can ask me, sir, a flow cytometer can measure how many proteins? So I give the answer. You can measure multiple proteins at a time on a cell, but for that you need a system which can measure multiple fluorescences. Very simple. For 5 proteins you can measure, but you have to use 5 monoclonal antibodies which are containing or which are tagged with 5 different fluorescent factors or 5 different fluorescent dyes. Okay. So if my flow cytometer is containing 1 laser, then generally we can detect 2 colors. 2 fluorescent color means we can detect 2 types of proteins. Then you can have 2 laser instrument, means 8 colors. 8 colors means you can measure 8 fluorescent dyes or 8 types of proteins simultaneously, or 8 type of fluorescent signals simultaneously. Then you can increase the number of laser, 3rd laser, then 14, then 18. So that depends upon you, right, what you can do. But there is a technical difficulty, and that difficulty also arises from a daily life observation. I tell you, all of you have seen a rainbow. A rainbow is theoretically having 7 colors. But practically when you can see the rainbow, you are not able to separate those 7 colors. You cannot clearly visible all 7 colors. Reason is very simple. These colors are having wavelengths near to each other, and when they have wavelengths near to each other, these wavelengths can mix with each other, and it is very difficult to separate the colors or analyze or observe the colors easily, right? So similar thing can come into flow cytometry. If you are adding 2 colors or 2 fluorescent molecules, it is fine. Maybe their wavelength will not overlap with each other, but adding 3rd color, you are increasing overlapping. 4 color overlapping, 5 color overlapping, 6 color overlapping, 7 color more overlapping, 8 colors. Imagine what will happen if you are utilizing 18 different fluorescent molecules used in same test tube. So it is difficult to analyze the data of complex experiments, because in complex experiment you try to understand multiple proteins at a time, or you try to understand multiple fluorescent signal at a time. And this spectral overlapping problem can be solved by a method which is called as compensation. So you can see here, if I am using these 2 colors, FIT-C and PE, this is the emission peak, this is the emission peak, but you can see that here you are having what? Overlap. So FIT-C is trying to enter into the spectrum of what? PE. If you are using these many dyes, 1, 2, 3, 4, 5, then you can see everybody is overlapping everybody else. So we use a mathematical method which is called as compensation. It is auto built in the instrument, but if you are having the knowledge of integration and partial differentiation, then you can also do the compensation. But nowadays we just use the software and it is done. I'm not going into the details of compensation, but I just wanted to tell you this is the problem, and here you require some mathematical knowledge, some voltage knowledge to solve the problem of compensation. But I just can show you one example. So here we have utilized 2 antibodies in our experiment. We have added antibody against CD4 protein, and you are having what? A label which is attached to these antibodies, and that is FIT-C. Then we have added antibody against CD19 protein, and which is having a label that is PE. So you can see you are having different population, one which is double negative, one which is only positive for CD19, and one which is positive for CD4. But you can see this population is entering into this quadrant. So falsely we are getting the information that there are CD4 T cells which are also expressing CD19, means we are having some CD4 T cells which are also the B cell. That is a wrong information. So we apply the compensation, and you can see that after compensation this population come down and you are having straightforward population, means now you are not having any spectral overlap. So compensation is the daily routine of flow cytometry people, and you can see if you are having 2 colors you can have the problem of compensation, but imagine you are having 8 colors, or imagine you are having 18 colors, 14 colors, how much it will be difficult, right? So here comes my second advice, and I will give you some numbers, some hard-hitting numbers, then we'll start our next concept. What happens nowadays that when we apply for industrial jobs, or we apply for PhD positions, we apply for good academic positions in research institutions and good universities, India or abroad, they ask you the question, what is your technical expertise? Okay. So technical expertise generally, I'm not saying all of you, but generally people say, even I was doing same in the bachelor's, generally people say that I'm good at RNA isolation or DNA isolation or plasmid isolation. But nowadays the problem is these things are not considered as your technical expertise. These are the routine things which anyways you have to do in a lab, whether it is industrial job or whether it is a research position, whether it is PhD, postdoc, whatever. Nowadays technical expertise means you are the expert of NMR, nuclear magnetic resonance spectroscopy. Then you are expert of mass spectroscopy, you are expert of flow cytometry, you are expert of real-time PCR system. So the advice is, it is your moral duty, whether you are in bachelor's or in master's, to learn one technique fundamentally, theoretically and practically, and you should be able to analyze the data. Okay. A master's student has completed the master's and applying for an industrial job, and he is getting the salary of, for example, 20K or 25,000 a month. Okay, but that person is writing in the CV and claiming that the person is very good in analyzing 8 color flow cytometry data. Then the same person get the job of 50,000. If the person say I am very good in analyzing the NMR data and mass spectroscopy data, then salary can go up to 75,000 also. That is the difference created by what? Technology. If you are applying for a PhD position in world's top research institution and you are saying you know only the DNA replication or DNA isolation, and some other person is coming and telling me that I can handle a mouse and I can analyze the single cells of spleen and thymus and lymph node of the mouse by utilizing multicolor flow cytometry, then who will be getting the position? Obviously the second guy. The world is moving towards more and more technological aspect of the biology. So it is moral duty for every person to learn one technique. I'm not talking that you learn multiple techniques, 10 things, 15 things, 50 things. Just one thing. But you should be having fundamental knowledge of that. You should be having experimental knowledge of that. And you should be having data analysis knowledge of that. And you know I'm doing flow cytometry now, I think 11 years. 11 years doing same technique and learning different aspects and making CV more and more strong. Getting my point? So first advice was you need the help of physics, chemistry and maths. And second advice is you need the help of a strong technique with you, which can separate you from the other average people in this world, or which can make you special. Everybody is looking for a special person. In Europe, they give you full scholarship to do PhD. But why they will hire you? You should be advantageous for their lab. And when you will be advantageous? When you are having fundamentals, good fundamentals with you, and one strong technique with you, which you can go and start from day one. Right? Start learning at least one technique. Now we move to second application. That is, we can analyze the live and dead cells. Very good, amazing. So how we can do? We can take single cell suspension, great, and we can incubate them with a dye which is called as propydium iodide, PI, which gives generally red fluorescence. And after putting the PI, we put the test tube in the flow cytometer. Then you can get this kind of graph where you are having x-axis PI fluorescence. So I can divide the graph into two parts. Here are the cells which are PI positive, and here are the cells which are PI negative. Okay. So generally more than 10 to the power 3 is considered as positive. So these are PI positive cells, one dot is one cell. And here are PI negative cells, one dot is equal to one cell. And the instrument is saying you are having 87 point something cells which are PI negative, and then 12 point something cells which are PI positive. Okay. So what is PI? PI is a DNA binding dye. It can go inside the cell and it can bind the DNA. It can bind to the DNA. It gives you the fluorescent signal after binding with the DNA. Great. It can do that. Now, the issue is PI enters only inside dead cells. So, a cell is giving you the signal of PI. It means what? That cell is dead because PI has entered and bound to the DNA. And if a cell is not giving you the signal of PI, fluorescence of PI, it means cell is the live cell. So PI positive are dead cells and PI negatives are what? Live cells. So within 5 minutes in a given solution, in a given cell suspension, you can find out whether your cells are PI positive means dead cells, or whether you are having live cells which are PI negative. Right? Next application, we can analyze the apoptosis. Very interesting thing. Really very interesting. So imagine a person is working on an anti-cancer drug. Right? And person wants to know that if he or she adds a drug to the cancer cells, then the cells are dying or not, and whether they are following the apoptosis pathway of the cells or not. How we can analyze it by flow cytometry? Very easy. Generally, a live cell, if you add the PI, then it will not enter it. And we can say a live cell is PI negative. And there is a protein which is called as phosphatidylserine, which is generally present inside the cell. And if the cell is entering into the pathway of apoptosis, then phosphatidylserine can come out, can come on the outer leaflet of the plasma membrane, and it can be recognized by a molecule which is called as annexin 5. Okay, so if your cell is live, then the cells will be negative for PI because PI is not entering, and they will also be negative for annexin 5 because the phosphatidylserine is present inner leaflet of the plasma membrane. So annexin 5 is not able to bind it. Okay, so we take the cells, great, and we stain them, we incubate them with two things, annexin 5 and PI. And after incubating, we put the test tube in the flow cytometer, and flow cytometer will be doing what? It will be measuring the fluorescence of annexin 5 and the fluorescence of PI. Again, we get a quadrant. On the x-axis, we are having fluorescence of annexin, and on the y-axis, we are having what? Fluorescence of what? PI. Again, one by one quadrant we can check. This is the first quadrant. Now we know that it is negative for x-axis, it is negative for y-axis. So I can say it is negative for annexine expression or annexine fluorescence, as well as it is negative for pi fluorescence. So a cell which is negative for both annexine and pi is a live cell. So software is measuring here and it is telling you are having 96% cells are live. Then we go to this quadrant. This quadrant is positive for x-axis but negative for y-axis. So positive for x-axis means it is annexine positive but pi negative. So what we can say? We can say this is early apoptosis. Early apoptosis means annexine has started to bind on phosphatidylserine, but cells are not completely dead because they are still pi negative or they are still y-axis negative. Then this is the quadrant which is positive for both. Means these cells are annexine positive and these are what? Pi positives also. So these are the double positive cells, right? Double positive means the apoptosis death is there. These cells are already dead because pi has entered in them, and since they are also positive for annexine, it means the reason of cell death is what? Apoptosis. And these are the cells which are pi positive, positive for y-axis, but annexine negative. It means they are negative for x-axis. So we can say they are pi positive, means they are dead, but the reason of death is not apoptosis. Why? Because if they are dead because of apoptosis, then they should be positive for annexine also. So live cells, only annexine positive means early apoptosis. Double positive for both annexine and pi means death because of apoptosis. And here you can see only positive for pi, dead but not because of apoptosis, means they are death because of some other mechanism like mechanical pressure, chemical pressure, or you can say that necrosis or whatever. Now the same cells, they have treated with small concentration of your drug. So you can see that now the live cells are 73%, 96 to live cells are decreased, increased more in apoptosis and increased in the death cell. Higher concentration of drug, you can see that live cells are very less, apoptosis positive, early apoptosis high, and dead are also high. So by utilizing this method, you can easily find out that what is the percentage of cells which are dying because of your treatment, because of your herbal extract, because of your chemical drug, and also whether the cells are dying because of apoptosis or because of some other kind of pathway, right? Easy method. You take cells today, you treat them with your drug. Tomorrow, within 30 minutes, you will be having the information whether cells are dying or not, cancer cells. If they are dying, what is the percentage of death? Yes, they are dying, percentage of death is known. Then what is the mechanism of death? What is the timing of death? You can do this experiment in time dependent manner also. You measure this thing after 12 hours, after 24 hours, after 36 hours, after 48 hours. So you can also find out that how much time it takes for your drug to kill the cells. Very interesting application. So we can find the proteins on the surface like CD3, CD4, CD19. So actually we are finding whether your cells are T cells, B cells, natural killer cells or not. Then we can find live and dead percentage of cell. Then we can find the live and dead percentage as well as apoptosis negative, apoptosis positive percentage of cells. So some really interesting applications are going on. Now, we can analyze the cell cycle of a system, right? So we are having a problem now. We are having a question now. The question is, you are given, for example, a certain number of cells, say 10,000, 50,000, 60,000, whatever. And I am asking you, please measure that out of these cells, how many cells are present in the G1 phase of the cell cycle? How many cells are present in the S phase of the cell cycle? And how many cells are present in the G2M phase of the cell cycle? Okay. And this I want to do by the flow cytometry because I want fast results, I want quantitative result, I want results in the form of numbers. I don't want Yes, cells are dying because in microscope they are this. I want, what is the percentage of cells dying? Here I want that in a given mixture of cells, out of 10,000, what is the percentage of cells which are present in G1? What is the percentage of cells which are present in S phase? And what is the percentage of cells which are presenting in G2 and M phase? Can we do that? Yes. For that, we are having some theoretical knowledge. Which kind of theoretical knowledge is? G1 is the growth phase. They have 46 DNA molecules. S means DNA replication is occurring, and G2 is DNA replication is already completed. So here we are having 92 DNA molecules double. Here we are having 46 DNA molecules. And in the S phase, we are having DNA molecules between 46 to 92. Because some cells will just start the S phase, some cells will be half at the S phase. Some cells will be having nearly DNA content of 50 DNA molecules. Some cells will be equal to 60 DNA molecules and all. So G1 will be giving you the DNA content equal to 46 DNA molecule because we have 46 chromosomes. G2 will be giving us the information of DNA content equal to 92 molecules because DNA replication has already been completed in the S phase. And S phase will be giving us the DNA content between 46 to 92. Now what we do the experiment, how we do the experiment? We take the cells, then we fix them. Fixing them means we use the chemical to stop the cells into whatever phase of the cell cycle they are. So imagine if you are having 10,000 cells and out of 10,000 cells, 2,500 in G1. So cells will be fixed in that position only. They will not enter into the S phase. Similarly, some cells will be fixed in S phase. Some cells will be fixed in what? G2 phase. Then we add the PI. Since cells are fixed, they are dead. PI will enter into the cells and it will bind to what? DNA. Okay. And we put this test tube into the flow cytometer, and flow cytometer will do its function. It will be doing what? It will be measuring the size of cell, FSC. It will be measuring the granularity of the cell, that is SSC, and it will be measuring the PI fluorescence. PI fluorescence means it is measuring indirectly the fluorescence given by DNA, because more DNA means more binding with PI, more red fluorescence. Less DNA, less binding with PI, and less fluorescence given by the PI. We put that and we get this kind of graph. Now we are not having a quadrant graph, but we are having a histogram, right? On x-axis we are having fluorescence of PI, which is actually giving us the information about DNA content. And on the y-axis we are actually having cell count. So if you can see, you are having three types of things coming here. One is the peak of G1. Second is the peak of G2. And this is the area of what? S phase. Now how to interpret this graph? It is very simple. G2 peak is having highest fluorescence, means highest DNA content. That can be what? 92 DNA. So these cells are showing you the fluorescence equal to 92 DNA molecules. This peak is showing the fluorescence equal to 46 DNA molecule content. And this area is showing you what? S phase. Means the DNA content between 46 to 92. Right? What software does? Software calculates area under the curve and tells you how many cells are G0 or G1. Here is the cell count. Here how many cells are in S phase. And here, how many cells are in what? G2 and M. So within 45 minutes protocol, you get the information about what? How many cells, what is the percentage of cells in a test tube is in G1? What is the percentage of cells present in S? What is the percentage of cells present in what? G2. Very easy to understand. Now, where I can utilize it? Very simple. Sometimes there are some anti-cancer drugs which kill the cells, and there are some anti-cancer drugs which don't kill the cells but they stop the cell cycle. Let's see what kind of result we get. So we have done one experiment. We have taken cancer cells and we have treated them DMSO, means no killing, it's just the control. Then we have treated them with a drug PD901, and then we have treated them with another drug, that is paclitaxel. So this is no death, no effect, no inhibition. So you can say you are having cells in G1, then you are having cells in S, and then here you are having cells in what? G2. So G1, S and G2. The system can calculate what percentage of each area and can tell you how many cells are in G1, how many cells are in S, and how many cells are in what? G2. Then PD901, you can say that in comparison to control, you are having very less cells in S and very less cell in G2. These two peaks are gone. So the conclusion is this drug is blocking the cell cycle in G1. So all the cells are collected in the G1. They cannot enter into S and G2. So most of the cells are coming at the fluorescent signal of what? G1 or 46 DNA. Here you can say that G1 is gone, S is also gone. Most of the cells are collected at what? G2. So you can say that paclitaxel blocks the cells into G2 phase of the cell cycle or G2M phase of the cell cycle. That is why whatever cells were in G1, they have crossed the S, they reach to G2, and after that they are not entering into next cell cycle because everybody is stopped at the G2 phase of the cell cycle. And it is well known fact that paclitaxel is a drug which blocks the spindle fiber formation by blocking the polymerization of tubulin protein. So obviously cells will do the growth, they will do the replication, but they will not do the prophase, metaphase and all these things because they will not be able to form the spindle fibers. That is why they will pass the G1, they will pass the S phase, as well as they will stuck into what? G2 phase, because they are not able to going ahead, they are not able to create two cells because they are not having normal spindle fibers. Very easy. Till now we have analyzed the presence of protein, then we have analyzed the presence of DNA, presence of live and dead cells, then we have analyzed how many cells are doing apoptosis, how many cells are doing necrosis, how many cells are live, and now we have analyzed that how many cells are in G1, how many cells are in S, and how many cells are in G2. Very nice, interesting experimentation, time saving experimentation and quantifiable experimentation. Why I like the flow cytometry? Because it can count, it can calculate everything for you. It gives the numbers, and numbers never lie. Here comes my third advice. Whenever you are reading a book, whenever you are reading a reference paper, whenever you are reading a reference book, always try to understand the graphs, because the results will always come, the experimental results will always come in terms of what? Graphs, in terms of numbers. So you should be good at reading and interpreting the graph. A biology student is not a good student if he or she is not knowing how to interpret the graph, how to say whether the graph is showing your favorable results or not favorable results, whether the drug is killing something or not. Everything, the final outcome of every biological experiment is in terms of numbers as well as in terms of graphs. So if we are really interested in becoming a scientist, a researcher, or we are really interested in making our career nicely in the biological research, then it is very important to understand graphs. Okay, so these were some basic applications. Now we are going to discuss some complex applications. But before that, I will be taking a break of 5 minutes. So in exactly 5 minutes, we will start again. You can have water. I am going to have some water, and then it is now 15:20. Exactly at 15:25, we'll start again. Thank you guys for your patience. Let's meet after 5 minutes. The most important application of flow cytometry is immunophenotyping. Immunophenotyping means analyzing the cells based on the surface markers and based on the CD molecules. So I do one experiment here again, and let's see how we can analyze it. So we are taking the blood. From blood we are isolating the PBMCs as we did last time, and from PBMCs we are adding the antibodies, like antibodies against CD3, CD4, CD8, CD19, antibody against CD25, CD44, CD33. Okay, we incubate the sample with these antibodies. So now how many proteins we are analyzing simultaneously in one sample? 4 plus 3 is 7. Very good complex analysis, right? And we will put the test tube in the flow cytometer. So I just tell you what these markers and proteins are signifying. So CD3 is present on T cells, CD4 presents on helper T cells, CD8 present on cytotoxic T cells, CD19 present on B lymphocytes, CD25 is present on highly on regulatory T cells or suppressor T cells, CD14 present on monocyte, and CD33 present on monocytes. Okay, now we are going to get the information about these many types of cells, and let's see what is the information. So first we'll get the graph between FSC and SSC. I have already explained you will be getting three types of populations: lymphocytes, monocytes, and granulocytes. Okay, then we will be selecting the lymphocytes, and out of lymphocyte we will be analyzing CD3 and CD19. What is Alexa Fluor 700 and Pacific Green? These are the names of dyes which are present on the antibodies which are binding on the CD3 and CD19. So here we are getting the information about how many are T cells, how many are B cells, and how many are negative. Now we move ahead and we select the T cells, and these T cells we analyze for how many are CD4 and how many are CD8. Okay, then we select these CD4 cells and we analyze them how many are positive for CD25. So I have analyzed lymphocytes, then in lymphocyte I have analyzed T cells and B cells, then from T cells I have analyzed helper and cytotoxic, then from helper I have analyzed how many are regulatory T cell. So I am going from lymphocyte to T cell to helper to regulatory T cell. So we are getting information about percentage of lymphocyte, percentage of B cell, percentage of T cell, percentage of CD4 helper T cell, percentage of CD8 cytotoxic T cells. Then within CD4 percentage of T cells we are getting the information about how many CD4s are actually regulatory T cells. Because CD4 cells, helper cells can be divided into further classifications like TH1, TH2, TH9, TH17, TH21, TH22, Tregs, regulatory T cells. So T cells are also of what? Multiple types, and we are doing it. And then on another side, from this area of monocyte, I'm saying how many are CD14 positive monocytes, how many are CD33 positive cells, monocytes are there. So this is a complex immunophenotyping. If I add more antibodies, I can analyze more type of cells. So everything depends upon how much complexity you are analyzing, how many antibodies you are adding, how many proteins simultaneously you are adding. If you are having knowledge of compensation, you can go up to 18 till now. So 18 is, I think, the limit of modern flow cytometers, where you can analyze 18 proteins simultaneously, mean 10 to 15 types of populations. Amazing technique, just done in half an hour, 1 hour's experiment. Obviously the analysis part is longer, but performing the experiment is easy and high throughput. Now, intracellular staining. So till now I have told you that you can add the antibodies which are binding to your surface proteins. Okay, and you can analyze them. What if your protein is present inside the cell? What if I want to analyze that whether a particular transcription factor is present inside the cell or not, expressing inside the cells or not? So the question is simple and answer is more simple. Yes, we can analyze them if we can send our antibodies, fluorescent tagged antibodies, inside the cells. And we are doing it routinely. We can take the cells, we can fix and permeabilize them. Permeabilize them means we are creating small pores. Then we can add the specific antibodies. So those antibodies will be entering inside the cell and will be binding to the target proteins. And here you can see that in this quadrant, I'm showing the cells which are FOXP3 positive. So what is FOXP3? FOXP3 is a transcription factor which is present in the nucleus of suppressor T cells. So I can also say that whether my suppressor T cells are expressing the transcription factor FOXP3 or not. Interferon gamma I am measuring here. Interferon gamma is a secretory molecule which is secreted from inside of the cell. I can do that. Getting my point? So by flow cytometry, you can analyze the external proteins present on the surface, but you can also analyze the proteins which are present inside the cells by just fixating and permeabilizing the cells and sending your antibodies inside the cell. After that, it's just the simple measure of FSC, SSC and fluorescent intensity. Very simple. We are following one very basic principle. Now, here comes my fourth. Oh, I will give the next advice after this. Now, application 6, one of my favorite. Right. Can we analyze the cancer stem cells? And people are doing it. And this is one of the landmark analysis by which the cancer research is progressing very fast now. So if I explain a tumor, so people say that cancer is uncontrolling growth of cells, uncontrolled cell division of cells. So you generate a tumor and the tumor is a heterogeneous mass of cells. Heterogeneous mass of cells means all the cells which are present in a tumor are not identical. Okay. There are some cells which can start new tumor and they have stem cell like properties and we call them as cancer stem cells. And there are remaining cells which cannot initialize the new tumor and those are cancer non-stem cells. They are not having the stem cell properties. So what happens if this is a tumor, if we give a conventional therapy to which the cancer stem cells are resistant, then what will happen? That even if few cancer stem cells are left after the surgery or after the chemotherapy or radiotherapy, they will generate a new tumor. Okay? But if we are having methods to identify the cancer stem cells, then we can give target specific cancer stem cell therapy. And then what happens? Even if you kill only cancer stem cell and remaining tumor will be there, it will regress very fast because there will be no cells left for the generation of new tumors or new mass of the cancer. Right? So it was very important to analyze the cancer stem cells. And now we are having so many markers which are cancer stem cell specific. So we take one example here. We say that breast cancer stem cells are known to have this phenotype. They are CD24 low expression and they are having higher expression of CD44 high. So we can analyze and identify the breast cancer stem cells as CD24 low and CD44 high. Right? So how we analyze by flow cytometry? Once we are having information about CD24 and CD44, then it is very easy to do. You take breast cancer tissue, you treat them enzymatically and mechanical grinding is there. You prepare a single cell suspension and you stain this single cell suspension with antibodies against CD24 and CD44 and you put them in the flow cytometer and you get this result. You have FSC and SSC, select the area of cell and on x-axis say you are having CD24 and CD44 on the y-axis. So these are double negative, these are double positive quadrant, these are positive only for CD44 and these are the positive only for CD24. So where will be my cancer stem cell? My cancer stem cells will be CD24 low, means they will be in this area and they will be CD44 high. So they will be having this quadrant. So this is the quadrant which is telling me, okay, these are the cells which are cancer stem cells. And I can find out that in a given sample or a given single cell suspension, there are 26.43% are cancer stem cells. Now, why it is important? Now, by using this method, if I am treating a drug, I am treating a cancer cell line or tumor with a drug, then I can find out whether the given drug is killing the cancer stem cells also or not. Because it is very important to find out new drug molecules and new therapeutic strategies where we kill cancer stem cells specific. So if they are dying, then from this area cells will be less. There is another marker which is called as ALDH. So ALDH is an enzyme, aldehyde dehydrogenase, which is expressed highly by the cancer stem cells and not by the other cancer cells. So you can see there are two famous cell lines of cancer. One is called as MCF7 and another is called as SKBR3. And you can see that there are very few cells positive for ALDH. So I can say that MCF7 is containing very less percentage of cancer stem cell. But you can see in SKBR3 you are having good number of cells which are positive for ALDH. So you can say that in SKBR3 there is a fair and good proportion of cancer stem cells. But in MCF7 there is not. So if I want to check the effect of a drug against the cancer stem cells, then obviously my choice of cell line will be what? SKBR3. My choice of the cell line will not be MCF7. So it is very important to know in the cancer research that which cells to target, how to analyze the cancer stem cells. And nowadays there are approximately 1000 to 1200 labs in the world which are working in the field of cancer research and mainly targeting what? Cancer stem cells. So if somebody is interested in doing research, further research in cancer or making career in the field of cancer biology, then it is very important to understand the concept of cancer stem cells, then concepts of EMT, epithelial mesenchymal transition, and the concept of metastasis, because these three things are going hot nowadays. Now, the cancer stem cell I am going to explain, I am going to tell you one very interesting story actually, or you can say one very interesting concept, analogy. So I like to explain the things by using some interesting analogies. So let's listen to that. So we see that in our normal life there is one issue, and that is the issue of illegal colonies or illegal areas where you are not allowed to make homes, but people are making that. People are making the slums. So in these illegal colonies, what happens generally? The people are very robust. Very robust, it means they don't have electricity. So they directly put a wire on the pole and they get the electricity. Less sanitization, but they manage that. So they are good managers. They learn to how to be robust, right? Similar situation is in the tumor. In tumor, the cells are very robust. If you stop their glycolysis, they will shift to something else. If you stop their blood supply, then they will shift to angiogenesis. So they are having methods to escape every problem. Now, how I can solve the problem? How I can solve the problem of slums or the illegal colonies? The problem was solved in the movie Nayak, Anil Kapoor. I am very inspired from Bollywood actually. So if you have seen the movie, then he has given the slum people new flats. So people went to the flats and they were happy. The illegal area will be cleared and then you can start whatever you want. So the similar approach can be utilized in the cancer, right? So giving new flats in the tumor means you take the cancer cells and you try to convert them into something else. If you are having tumor in kidney, then you can design a therapeutic system where you can convert the kidney tumor cells into nephron, or skin tumor cells into keratinocytes, or breast cancer cells into breast epithelium or fats or in adipocytes. And people call it as translational therapy or differentiation therapy, not the translational, but differentiation therapy, where you can convert the cancer cells into normal cells. Right. Now, what is the problem with this approach is, in slum people or in illegal colonies, there are some guys which are very stubborn. What they do, if you give them new flats, then they give their flats on rent, they get money from that, they come back to old place and they start new illegal thing, right? Similar thing is happening in tumor. You can convert any cell into whatever you want. You can kill any cell whatever you want. There are some cells which are very stubborn, and these stubborn cells are what? Cancer stem cells. So it is very important to target the stubborn cells or stubborn people. Right? If you do that, then regression of the other system works very fast. And this was a very interesting observation which I found, that you can make a good analogy and you try to understand the concept. And here comes my fourth advice. The fourth advice is, a good biology student should be an amazing observer. Amazing observer means, I ask you one very simple question and you give the answer to yourself. Everybody has learned Mendelian genetics. Tall plant, dwarf plant, red color flower, white color flower, smooth seeds, then rough seeds, and so many things. But if you go from your college to your home, whether by bus or whether by bike, whether by on your foot or on your feet, whatever you are doing, how many of you actually observe that in the way or on the way, how many plants are actually dwarf, how many plants are actually tall, whether they are following the Mendelian genetics ratio or not? The answer is nobody, or answer is very few people. For us, biology becomes a subject. We don't try to connect it with our real life. We don't try to observe the biological phenomenon in our real life. That is why every time we have to remember the things. People come to me and they ask me the question, हम बहुत भूल जाते हैं. Sir, we forget the things very easily. I always tell the people, you forget the things which you remember. आप उन्हीं चीजों को भूलते हो जिसके लिए आपको याद करने का स्ट्रगल करना पड़ता है. You are not going to forget anything which you are actually observing and loving. And once you start loving these things, then you will start observing and making analogies, making good connections, making some funny methods of your own to understand the things. Then the subject, then the complex things of biology will become very easy. And to be a good scientist, you need fundamentals, you need technology, you need amazing observations, and you need something extraordinary by which you can explain the biology very easy. I like to make so many analogies, so many. If somebody has watched a movie, Sholay, in Sholay, what Gabbar Singh did, he captured the Dharmendra and उसने धर्मेंद्र को दो पोल्स के अंदर बांध दिया, एक हाथ इधर एक हाथ इधर, राइट. एंड फिर उसने बसंती को बोला डांस करना पड़ेगा अदरवाइज ये मर जाएगा. सो इफ यू लुक एट द पैटर्न of Dharmendra bound with those two poles, then the exactly similar structure is found in the muscles where the poles are equal to myosin molecules and Dharmendra is what? Actin. So if Dharmendra pulls the poles towards him, the muscles are contracting, and Dharmendra is letting loose the poles, and muscles are what? Relaxing. Amazing analogy. There is a song, जहां मैं जाती हूं वहीं चले आते हो, यूं तो बताओ कि तुम मेरे कौन हो. If I ask which biological situation this song will fit perfectly, is it a easier funny way to remember it? Yes. In prokaryotes, the transcription and translation are coupled with each other. RNA polymerase move along the DNA template to make RNA, and the translation is coupled, so ribosome move just after the RNA polymerase on the mRNA to create what proteins. Best example, RNA polymerase is telling the ribosome, jahan main jati hu wahin chale aate. Analogies, observation, loving the subject. That is how you become a successful student. And I always tell the people that you should be a good biology student. Other things like career, money, and all things, these things will fall automatically in your lap if you are really interested in doing something. And to be interested, first advice, second advice, third advice, and now you should be amazing observer. Now let's move to next application. Can we use flow cytometry somehow in plants? Yes, it's very simple. So we have already done the cell cycle where we have seen that the PI can bind the DNA, can bind to the DNA and can give you the signal, and based on the signal you can find out the amount of DNA content. Similar thing you can do here. In plants, we know that plants are not generally 2N. They are having different ploidy levels, 2N, 4N, 5N, 3N, triploid, tetraploid plants. And it is very important to check their ploidy level for different purposes of plant tissue culture and in vitro growth and all. Can we check the ploidy level in plants by utilizing flow cytometry? Yes. We take the leaf tissue, we isolate the nucleus and we add the PI. So if you are having only 2N DNA, on y-axis we are having DNA fluorescence, which is given by PI. If you are having 2N DNA, you will be getting some cells. Then you are having 4N DNA, then you are having 8N DNA. More the DNA, more fluorescence given by what? PI. And the software will calculate for you that what is the percentage of cells in plant present in 2N stage, 4N stage and 8N stage. Very widely used method, a very widely used experimentation protocol of flow cytometry in plants. Now something new. This. What is this? One of the complex experiment, one of the most complex experiment in flow cytometry. So if I ask you that I am giving you serum or I am giving you a supernatant of cell culture and I tell you quantify the amount of interferon gamma in this given serum, what method you will use? ELISA. ELISA is used to measure the proteins present in a liquid. Now we are not talking about the cells, we are talking about the proteins. Okay, can we measure the proteins by flow cytometry in a given serum or in a given liquid where cells are absent? Yes, people will do ELISA. Then I ask the same person, you take interferon gamma and TNF alpha, can you do both of them for me? The person will say yes. But for that we will require what? 2 ELISA kits. If you want to measure third cytokine, 3 ELISA kits, 4 cytokine, 4 ELISA kit, 5 cytokine, 5 ELISA kit. While ELISA will take 10 days to do optimization and all. Imagine you want to measure 50 cytokines. For that you will be requiring 50 ELISA kits. Very expensive, very time taking. Flow cytometry comes with a bead array, cytokine bead array, in which 13 to 18 cytokines or 13 to 18 proteins without cells can be measured in a given liquid, and the sensitivity is the instrument can measure up to 2 picograms per ml. Amazing. You can see that we are measuring IL-10, 12, 17, alpha, gamma, alpha, GM-CSF, IL-4, 6, 5, 2, 9. Each circle is denoting one type of cytokine, and you can measure their fluorescence intensity, and within a day you can do 100 samples. That is the speed of instrument, and each sample you are measuring 12 to 13 cytokine, so you are finishing your research paper in a day. Amazing technique. High throughput analysis. You can measure cytokines, you can measure chemokines, you can measure growth factors. Whatever is secreted in terms of protein by the cells can be analyzed by CBA or bead array, flow cytometry based bead array system. the last thing of today's lecture. It is our experiment. And I personally like this experiment a lot. So imagine you are working on a drug which is anti-malarial, and you are saying, by my drug the plasmodium will die, or you are working in plasmodium will die, right? So how you are going to do that? You culture the plasmodium, will put the drug on the plasmodium culture, and then you will see whether they are dying or not. But actually by flow cytometry you can analyze whether the plasmodium is dying in its natural habitat or not. And what is the natural habitat of plasmodium? The RBCs. So what we do? We take the RBCs, we infect the RBCs with plasmodium, and we put a DNA binding dye. What will happen? RBCs don't have nucleus, so they don't give the fluorescence for DNA binding dye. But if RBCs are infected with plasmodium, they will be giving the fluorescent signal, because now the RBCs are not giving the signal, but the plasmodium is present in the RBC. It is having the DNA and it is giving the signal. So you can see here, this is the RBC peak and this is the fluorescence intensity, and you can see there is no peak of that. So RBCs are not giving you the signal. Now you have infected the RBCs with the pathogen. The pathogen is going inside, and you can see there are some cells. How many cells are infected now? 10.98. Then you treat the cells, and then you can see the infection goes from 10.9% to 5.8%. So here we are testing a drug against plasmodium, not just by treating the plasmodium in a culture plate, but in its natural environment, which is giving us more authentic results. And what was the out of the box thinking in the complete process? That RBCs don't have nucleus, so they don't give a DNA content fluorescence. But if a pathogen is present in the RBC, it will start giving the signal. And once you are having the signals, you can measure it, and you can measure the percentage infection or intensity of infection. Amazing experiment. What are other applications of flow cytometry? People count CD4 T cells in the HIV infection diagnosis. They do immunophenotyping for the studies of different blood cancers. People are measuring the number of different type of phytoplanktons based on their autofluorescence. People are using food safety and the flow cytometry. People are utilizing flow cytometry in analyzing different type of adult stem cells, embryonic stem cells, and induced pluripotent stem cells. People are also doing sperm quality analysis by flow cytometer. We can measure mitochondrial membrane potential by flow cytometer. We can analyze different signaling pathways. They are using plant cell analysis. They are using circulating tumor cell analysis, and so many applications. And somebody asked me, what are the limitations of flow cytometry? Then I always give this line: imagination is your limit. Imagination is your limitation. If you can imagine one experiment, and if you are having cells and protein information with you, you can analyze anything on flow cytometry. And here comes my last advice. One of the most and one of the funniest and one of the most interesting advice is, a good biology student is an out of the box thinker, and he or she can give most of the answers of normal questions also by using the knowledge of biology. In different institutes, when you apply, there are sometimes group discussions, and I was sitting in a group discussion, and my topic of the group discussion was, God is male or female explained by molecular biology. My friend was having one topic, honesty is the best policy explained by biochemistry. Once somebody asked me, what do you prefer, hot coffee or cold coffee? The normal people who are not able to use biology as their weapon or science as their weapon. They will be saying, I prefer cold coffee because of this taste and hot coffee because of the taste or energy or whatever. But what answer I will give? I prefer hot coffee over cold coffee. Reason is very simple. There is a concept which is called as surface tension. So surface tension is a physical force which is going to explain the spread of a liquid on the surface. So if surface tension is high, then a spread of a liquid is less, and if surface tension is low, then the spread of a liquid is high. So surface tension also depends upon the temperature. If you increase the temperature, the surface tension goes down. So why I like hot coffee? Because it is hot, so it is having less surface tension. It is having less surface tension means it can spread more. So when I take a sip of hot coffee, it spreads more on my tongue, simultaneously activating more taste buds. So I get a better taste of what coffee. In cold coffee, it is a cold liquid. It is icy liquid. Surface tension is very high. More high surface tension means very less spread on the tongue. So I'm not able to activate more and more taste buds on my tongue. That is why I don't like the taste of cold coffee. I am giving the answer in terms of what? Biology. Right? शंकर भगवान ने जहर पी लिया था और अपने थ्रोट में रोक लिया था, इसोफेगस में, फिर भी वो क्यों नहीं मरे? If somebody asks me to make a biological hypothesis, then I will say he didn't die because he took myogenic toxin. So you have neurotoxins, then you are having myogenic toxins. So sorry, he took a neurogenic toxin or neurotoxin. So neurotoxin acts only on neuron, and he stopped the toxin in his throat or in the esophagus. Esophagus is having very less connection with neurons, mostly made up of muscle. So he didn't have any drastic negative effect. So he was knowing the biology. Imagine you are sitting in interviews and you are giving the answers of everything in terms of biology. What kind of impression it will leave? And it will really, really separate you from other average students. And that is why we are trying to make these videos and we are keeping these webinars to explain the biological techniques and different complex biological things. So more and more people get inclined towards biology and try to make an amazing, successful career in the field of what? Biological sciences. So thank you guys. Thank you very much for your patience and your time. So tomorrow we will be discussing on animal cell culture, or mainly human cell culture world. What are the misconceptions, what we do, what should we not do, and as well as the real-time PCR. Now guys, I am ready to take some questions. You can type your questions in the chat box and I will try to give the answers. Anything you can ask from today's session as well as other biological questions, and I hope you liked the sessions.